Hello, welcome students. We shall continue the chapter Biomolecules. In the previous video, we have studied that the molecules which we extract from any living is called biomolecules. You have already seen that when any livings get that when crushed and mixed with trichloroacetic acid and then the filter in the filtrate, that is acid soluble pool, and the unfiltered, that is called acid insoluble pool. So those we have studied, and in that we have seen that in the acid soluble pool, those elements whose molecular weight is in between 18 and 800 that we get, and above 10,000 Dalton's molecular weight, these compounds do not filter, they on the acid insoluble pool. Understand? Means in that we get amino acids, fatty acids, monosaccharides, nucleotides. It is these we get. Amino acid, as already I have told you, that it is a derivatives of methane, that is COH, means having COOH and NH2 group. These both are ionizing property, and that I have already told you that in the ionization positions, both amino and carboxylic groups is in the ionizing positions, and that is called Jutter's ion. As it is already discussed in the previous video. Now the fatty acids, we know that fatty acids is the actually lipids. Lipid is the composing of, it is the fatty acids plus glycerol, and fatty acids, this is the the general formula of the fatty acid, CH3, CH2ON, COOH. This is the general formula of fatty acid. It is of two types. One is saturated, next one is unsaturated. If only single bond between two carbon is found, that is called saturated, and if between two carbons at anywhere double or triple bond is found, that is known as unsaturated. Now the monosaccharides, you know that these are the carbohydrate products, and that its general formula is CH2ON. It's the monosaccharides. Mono means one, having one saccharide. The compounds may be C3H6O3, that is triOH, C4H8O4, that is tetraOH, C5H10O5, that is pentose, and C6H12O6, that is hexose. It is an example of glucose, understand? And monosaccharides, having one single unit. When two monosaccharides combines to form, that will form disaccharide. As for examples, the sucrose, it is made of glucose and fructose. Understand? That is pentose and hexose. The structure of pentose we make. HOH, first, there are the C carbon, C, first position, second, third, fourth and fifth, that is C5H10O5. This is the position of pentose. And the hexose, first, there HOH. So first, second, third, fourth, fifth and sixth. This is the structure of glucose, and all the HOH are found. Similarly, all H here. So this is the structure of. So it is the hexose and it is the pentose. Pentose, in the nucleotide or DNA, we shall see that two type of pentose sugar we get. One is deoxyribose and next one is ribose. About that we shall discuss at the time of nucleotide. Monosaccharides, when one monosaccharides, that is this general formula CH2O, that is this one. When two monosaccharides will combine, that will form disaccharides. When the number of monosaccharides is 2, that is disaccharides. Number is 3, that is trisaccharides, and many polysaccharides. Many, that is polysaccharides, and once about 3 to 9, they all are come under one group, that is called oligosaccharide. So monosaccharide, disaccharide, oligosaccharides and polysaccharides. So these are the polysaccharide is the starch. About this we shall discuss in the next video. Now the nucleotide. Nucleotide is composed of nucleosides plus phosphoric acid, that is phosphate, and nucleosides is composed of nitrogenous base and sugars that is combined to form nucleoside, and the nucleosides is composed of purine and pyrimidine. These are composed of. Now in the purine we get adenine and guanine, and in the pyrimidine we get thymine, cytosine and uracil. These are the bases of the nucleotides or DNA. This is a double layering structures. And this is a single ring structures. Understand the main difference between that? That is adenines and guanine. These are double ring structure, and that is a single structure. When this adenine or guanine and the base will be formed A, T and G and C, that is in the DNA or deoxyribonucleic acid, and in the ribonucleic acid in place of thymine there is uracil, and shortly we described ATGC. Now this ATGC, they when combined with the sugar, means if that is deoxyribose in DNA, if it is ribose in RNA, when this combined with phosphoric acid, that will form nucleotide. These two nucleoside, and when nucleoside is combined with phosphate or phosphoric acid, is called nucleotides. And when many nucleotides are combines, that they forms nucleic acid. So these are the some compounds which we get. Now the next point is metabolites. You know the metabolism. It is the sum of anabolism plus catabolism. In it, something is formed that is synthetic, means constructive, and in which the some big compounds splits, that is destructive. यहाँ कुछ चीजें बनती हैं, synthetic होता है, यहाँ कुछ टूटती हैं, that is catabolism. So whatever the product of metabolism, either intermediate or final, that is called metabolites. Means products either intermediate or final of metabolism is called metabolites. Understand? Whatever the products, intermediate means during the chemical reaction some compound forms, that is called intermediate, and some finally that is formed, that is final product. So whatever the product, that is called metabolites. Now metabolites is of two types. One is primary metabolites and second one secondary metabolites. These metabolites are directly contact to the lipids, means they are directly related to the growth and development of the livings, and these are not directly but they are indirectly or necessarily related with the growth and development. That is, we can say that those metabolites which are directly related to the development and growth of the living, and that is not directly but indirectly or accessory, so these are called secondary metabolites. In the primary metabolites, the protein, nucleic acid, polysaccharides, means all the constituents which we have studied in the last table, that is hydrogen, nitrogen, oxygen, they all are directly related to the growth and developments of the living. Protein is the development of cell membrane and cells. Nucleic acid is the genetic materials, and the polysaccharides gives energy or strength. They are directly related to that. But secondary alkaloids, flavonoids, essential oils, scent, gums, spices, although they are not directly essentials, but they have some economic importance also. So they are the products of the metabolism, and so they are known as secondary metabolites. Understand?